Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 39, where I will cover the diversity of sesquiterpenes, diterpenes, triterpenes and polyterpenes. I have mentioned about these terminologies in one of the previous classes. So, what I am going to cover, we will cover briefly the biosynthesis of sesquiterpenes, and then we will see the product diversity of sesquiterpenes. Then we will see the product diversity of diterpenes and triterpenes, and finally, I will give you a brief outline about polyterpenes. So, let us go to the board, that is sesquiterpenes. Biosynthesis and product diversity of sesquiterpenes. So, sesquiterpenes are C15 compounds, which I have already mentioned in one of the previous classes, and it also there is an enzyme called sesquiterpene synthase, and the substrate is farnesyl pyrophosphate. So, farnesyl pyrophosphate is formed from DMAPP, dimethyl allyl pyrophosphate, and the enzyme will be farnesyl pyrophosphate synthase FPPS, and which requires 2 molecules of IPP, 2 into IPP. So, that means DMAPP is C5 and 2 IPP means 2 into C5. So, total 15 carbons, leading to the formation of this FPP skeleton, which will act as the substrate for sesquiterpene synthase. I just use the it should go down up to this much, come here, go down, go down. So, this is basically the structure of farnesyl pyrophosphate FPP. I will write the full name here. Or farnesyl diphosphate. If we are N, okay, N is missing. Sorry. Okay. This will be converted into different sesquiterpene molecules. So, here I am not discussing the reaction mechanism. So, I will first put the several products which form from farnesyl pyrophosphate. So, one main example is your farnesene. So, farnesene structure I will draw it here. If which is pretty similar to FPP. This is alpha farnesene. So, apart from this, another major structure which is of common occurrence is beta caryophyllene. So, this is the problem. This is the structure of beta caryophyllene, beta caryophyllene. So, and another representative structure I will draw here, which I will draw as bisabolone. This is alpha bisabolene. Okay. Any more interesting one is, another interesting one is humulene. I can also put it. And jarmacrine I also need to put it. So, these 5 structures only I will show. One is jarmacrine. Okay, so let us draw the humulene. HUMULENE, and finally, I will draw the structure of jarmacrine. So, the jarmacrine this is the structure of jarmacrine. So, these are all volatile compounds and you will find it in different plants. Alpha farnesene you will find in so many plants, particularly you will find in the Rutaceae plants in the orange family. Caryophyllene also you will find there, you will find in the Docus carota, and bisabolone you will find in several species including the santalum. Jarmacrine also is a common sesquiterpene which is also found in different floral scent, and humulene is specific for the hops. So, in the hops flower you will find humulene, and the product diversity is much more in sesquiterpenes than that of monoterpenes, although formation is a bit more complex. Okay. Now, of course, there are these are all the volatile sesquiterpenes, but apart from that there are several non-volatile sesquiterpene also exists, and that act as defense compounds, and it accumulates in different plants in different tissues. So, that also I will list it up, but before that okay, let us go to the next slide. So, here what I am going to show that from GPP from FPP, that is farnesyl pyrophosphate, there are different cations generated. For example, one is germacryl cation, which contributes to germacrin and related compounds. So, that I am going to show you. So, from germacryl cations different product formation occurs. For example, from germacryl cation, apart from germacrin A, which is GERMA CRENA, germacrin A, germacrin B and germacrin C are also produced. Germacrin D are also produced. C I am not showing. So, apart from germacrin A, both germacrin B and germacrin D are produced by different rearrangements of the skeleton which originated from germacryl cation. So, from germacrin A, the pathway contributes towards the formation of epiaristolochine. And that finally lead to the formation of capsidiol. Whereas, from germacrin A, two more root originates, one in this way, one in this way. The short root contributes towards the formation of parthenolide. Whereas, the other root from here guaiacyl cation is produced, GUAIYL, guaiacyl cation is produced, and from there it produces martesine, and from martesine chamazulin is produced. Now, martesine and chamazulin basically are the component of Marticaria chamomilla. So, the scientific name of the plant is the English name is German chamomile. So, the scientific name is Marticaria chamomilla, which is called German chamomile or German chamomile. So, the English name is chamomile. Okay. And from FPP another cation is formed, which is germacryl cation. Okay, germacryl cation I have said. Or germacryl cation also contributes to pactolol. So, germacryl cation also contributes to pactolol. So, I will put another arrow here, which will make PATCHOULOL. So, this is basically a tricyclic sesquiterpene, and this is very important because it is used as perfumery raw material. And it is also the major component of patchouli oil. This is Pogostemon cablin. This is the scientific name. Okay. Now, from FPP another cation formed, which is called bisabolyl cation. Bisabolyl cation, and from bisabolyl cation two different routes originate. One contributes to bisabolol. So, I put it alpha bisabolol, BISABOLOL, and that eventually formed bisabolol oxide A, bisabolol oxide B. Alpha bisabolol oxide A and alpha bisabolol oxide B, whereas the other route contributes towards the formation of artemisinin. So, I put the arrow in this way. So, this makes first amorpha 411 diene that subsequently converted to artemisinic acid. Sorry, again problem with the pen. Acid and artemisinic acid, artemisinin is produced. artemisinin, artemisinin. So, put the arrow. Now, artemisinin is basically a Chinese medicine obtained from the plant which is called Artemisia annua. Sorry, I have to use the different color. Artemisia annua. This is the English name is called sweet wormwood. The Chinese name of artemisinin is King Haosu. So, we will not write that, it is such a difficult to pronounce. So, this is from FPP different cations formed as a result of this farnesin synthase reaction. One major is this germacryl cation, another one is bisaboyl cation. From the germacryl cation, we will see the formation of germacrin, parthenolide, capsidiol, martesine and chamazulin. So, chamazulin and martesine are the component of this marticaria essential oil, whereas capsidiol is basically a phytoalexin compounds which accumulated in the plants to protect against fungal attack. So, okay. So, it is okay, I will talk about that now in the next slide. That means other sesquiterpenes which plays important role in defense. So, or the compounds that basically plays important role in defense. So, one is because I will not draw all the structure. One is okay, if we start with FPP, farnesyl pyrophosphate. So, start from this side FPP. From FPP, one root contributes to the formation of epiadistolecin, epiadistolocin, epiadistolecin. So, from epiadistolecin capsidiol is formed, capsidol is formed. So, capsidol and epiadistolecin synthase enzyme contributes to the formation of this product epiadistolecin synthase, that is nothing but a part of sesquiterpene synthase. And so, what I have mentioned you can also note it down here, this is basically a major phytoalexin and it is found in pepper and tobacco. Phytoalexins are those compounds when plants are healthy you will find very in trace amounts, but when plants are under attack then the machinery becomes active and leading to enormous amount of synthesis of these compounds. I have mentioned this in one of the very early classes that the three ways these defensive specialized metabolites are formed. Okay. Other route from here, which contributes towards the formation of delta cadinine, cadinene. So, this is found in cotton. This is found in cotton, and this finally makes the structure which is called gossypol. So, gossypol structure I will draw. So, the gossypol GOSSYPOL. What is gossypol? Gossypol is a sesquiterpene dimer, sesquiterpene, sesquiterpene dimer. So, let me draw the structure. Okay, the gossypol here. So, gossypol structure is very interesting. 4 benzene ring, 1, 2, gap, OH, CHO, OH, so this is the structure of gossypol. So, now another defensive sesquiterpene, which is called lubimine. Lubimine, LUBIMIN, which you will find in potato. So, and lubimine is basically formed from veti. Vetis spiradine, and vetispiradine is basically produced from FPP by this vetispiradine synthase. So, these subsequently converted into lubimine. So, this is so that means lubimine is also a defense compound which accumulated in the potato. So, I have said the different defensive sesquiterpenes that accumulate in different plant. Similarly, in Docus there is a daucine. Daucine is a sesquiterpene which accumulated in Docus. So, I can write it here. Daucine accumulates in Docus, that means carrot. In ginger also there is another sesquiterpene which accumulates, which is called gingiperine, PERENE. So, this is found in ginger. Okay. So, with this information, now I will go to the very briefly about diterpenes. So, diterpenes are basically C20 compounds. So, that means diterpenes are formed from DMAPP with addition of 3 molecules of IPP, and the enzyme is GGPP synthase, GGPPS, S stands for synthase, okay, GGPP synthase, leading to the formation of GGPP. So, GGPP only structure I can draw because of shortage of time. I will not draw the other structure, but GGPP structure is pretty complex. Let me try. So, this is the structure of GGPP. GGPP stands for geranyl pyrophosphate. This acts as the substrate for the subsequent enzymes, and this leads to the formation of different diterpene compounds. For example, I just write the name of the compounds. One is called abitadine, ABIETADINE. Abitadine. So, from abitadine the abitic acid is formed, ABIETIC, abitic acid is formed, and the enzyme is abitadine synthase, AB synthase. Similarly, one route contributes towards the formation of flowball, PHO floorball, BOL floorball. So, this is an ester found in the latex bearing plant, particularly euphorbiaceae. Okay. Another one is basically the casbane, CASBENE. Let me make it clear spelling, otherwise you will not. CASBENE. Casbane, what is casbane? Casbane is again a phytoalexin. It is a defense compound and it is found in castor bean, which is called Ricinus communis scientifically. GGPP also contributes towards the formation of taxadiene, Taxadiene. So, it is basically contribute to the skeletal of the taxol. So, the taxadiene synthase is responsible for that. Taxadiene synthase, and GGPP also contributes to the formation of geranyl geraniol, which is a common volatile metabolite found in different flowers. Okay. So, this is in brief about the different diterpene compounds. Now, if I go to briefly on the triterpene compounds, triterpenes. So, triterpenes are basically C30 compounds that also originate from two molecules of FPP that ultimately formed squalene. Squalene is considered as a precursor of triterpenes. So, the enzyme is squalene synthase, which requires two molecule of FPP. So, squalene is again the C30 compounds, and from squalene different compounds originate like cucarbitacin E. Okay. From squalene oxidosqualene is formed, SQUA. Oxidosqualene is formed, and from oxidosqualene different compounds originate. For example, cycloatenol, from there brasinolide is synthesized, and from oxidosqualene another route contributes towards the formation of beta amyrin. And from beta amyrin oleanolic acid is formed. So, just to tell you the product diversity, squalene also contributes towards formation of other sterols, for example, cholesterols, and also contributes towards the formation of stigmasterol and cytosterol. Sigmasterol and cytosterol structures are pretty similar, only this contains a double bond which is absent in this structure. Okay. And steroid molecules with an alcohol group is sterols, and these sterols also, one such sterol is phytoecdysone. So, this accumulates in the plant and the insects lay eggs, the larva are formed, then larva to pupa conversion, then when pupa basically they when the larva consume the phytoecdysone, the pupa formation get hindered because of its toxicity. So, this has the steroid structure. Apart from this, there are two important compounds which comes under triterpenes, which is ammonin, YAMONIN, which is a type of saponin, and another is well known digitoxygenin, digitoxygenin, which is a cardenolide. That is at low concentration, it has a role in protecting the heart from other diseases. Okay. So, this is about the different triterpenes which accumulate in different plants, and they play important role, for example, the protection of the plants, protection of the membrane structure, and different aspects. So, this is about the triterpenes C30. C40 will be these carotenes, which I will cover in a separate class. Now, okay, maybe I should stop here, and in the next class when we will talk about carotenes, before that I will briefly talk about the polyterpenes and oleoresins before I move into the carotene structure. So, with this I will end this class. Thank you very much.